Hi, welcome back. This is Dr. Hadi here. Today's topic is from the biochemistry, advanced level topic about the classification of enzyme. Okay, we will discuss today the different systems, the different naming system given to the enzyme. Well, the first thing is why there is a need to classify enzyme. Why we give name to enzyme? The answer is very simple. In order to get something easily, in order to understand things easily, in order to get the idea clearly, sharp, we need to classify them. You know, in different subjects we classify objects. In biology we classify animals, we classify different compounds. We always perform classification in chemistry, in physics, and in other subjects just for the convenience, so that students get the idea very clearly. So enzymes must be classified and their classification should be easy. So in the beginning, the enzyme classification was non-systemic or improper classification. In the beginning, the classification was improper, and you can also call non-systemic. What is non-systemic? I will explain, and later on enzymes were given a proper classification system. I will also explain this with you. So there are two ways by which enzymes are classified. The first one, the conventional one, is the improper classification. It's very interesting, that I will tell you now. Here is by adding the suffix ase to the name of the substrate or to a word. Means, you know, enzyme act on something. Every enzyme that we see, they act on something. Okay, so if enzyme act on a substrate, let's suppose the substrate is a sucrose. This is what? This is the substrate. This is our substrate, sucrose, and the enzyme which is acting on the sucrose will also be given the name suc R. Now after R we will not add OSE, sucrose. We will just remove this O and we will add the word ASE. This word which comes at the end is called suffix. When a word comes in the beginning, it is called prefix. In the beginning, jo shuru mein aa jaye, which comes in the start, it is called prefix. Jo baad mein aa jaye, which comes after the word, that is called as suffix. So this is our suffix ASE. Now this is the name of the enzyme. Sucrase is what? Sucrase is the name. Sucrase is the name of the enzyme. Now which enzyme? Any enzyme that work on the sucrose. So the name of the enzyme is according to the substrate on which it act. So on sucrose, sucrase will be there. On lactose, lactase will be there, right? So this is one system for the naming of enzyme. That is very old system of giving names to the enzyme. The second thing in the same is either you add the word ASE to a word or a phrase describing the activity. Now this time we will get some activity, like there is an enzyme and the enzyme is involved in oxidation or hydrolysis. Now what is oxidation? Oxidation is a chemical process in which a compound loses oxygen, hydrogen, or it gets oxygen. This is called what? Oxidation. Okay, so those enzymes which are involved in the process of oxidation, they are called oxidase. They are called what? Oxidase. So again we are putting what? We are placing ASE as a suffix, because this is the activity. Oxidation is a chemical reaction, it is a kind of activity. So the enzyme which are involved in this activity, they will be called oxidase for oxidation. And hydrolysis is another activity. Hydrolysis is another activity in which some compound, something is broken down by the help of water. कोई चीज पानी की मदद से टूट जाता है. Hydrolysis. So the enzyme which is involved in that activity, they will be given name what? Hydro. Now not lysis, hydrolase. So if you notice here, we are adding the word ASE after the name of the sucrose, after the name of the substrate, or after the name of the activity. So in both the case, we are adding what? We are adding ASE. So this is a trivial kind of system to give names to the enzyme. So we have a name. This hydrolase is the name of enzyme, oxidase is the name of enzyme, sucrase is the name of enzyme. Fine. This is one thing. Now come here. Next is name by discover for a broad function. Now there is some broad functions. There are some broad functions like digestion. Look, digestion is a broad function. In the process of digestion, many enzymes are involved. So in the beginning, when there was no proper system to classify enzyme, in the beginning they started giving name according to the function. For example, the word pepsin was given to the enzyme. Yes, the pepsin is the name of the enzyme. This we are agree. Why this name came? How this name came? Because the word pepsin come from a Greek word pepsis, and pepsis means digestion. Because this enzyme was involved in the process of digestion. Because of digestion, the name of enzyme was given as pepsis. In the Greek, pepsis means digestion. So pepsin and pepsin. This is not a very highly specific name. This name is given because of this digestion process. So you can say that the name of the enzyme is called digestion enzyme. In Greek it is called pepsis. Now we call it pepsin. So this is another way of giving name to the enzyme. I will tell you what were the problems that came into that hole, and because of which we rejected all this system, and we got another systemic way to classify names. Okay, so this was the second thing. The third one is the naming by their source. Some enzymes were given name according to the source. For example, there is an enzyme that we call it trypsin. This one, trypsin, is another enzyme. Why this name was given as a trypsin? Because this trypsin word comes from a Greek, that is triene. And triene means if you rub something with other thing, this is called triene in Greek. Because this enzyme was obtained by the process of wear. When you rub the pancreatic tissue with glycerin, you have pancreatic tissue and you have glycerin. When you mix them, and as a result of this you get some enzyme, that enzyme will be called as triene in Greek, and from this triene we got the name trypsin. So this is the source where the enzyme came from. The enzyme came from by rubbing that pancreatic tissue with the glycerin. This is the source, and because the enzyme came from this source, we gave the name according to the source. So in the non-systemic or improper classification, the enzyme were given name in three ways. Number one, by adding the phrase, the word ASE, as a suffix to the name of the substrate or according to the activity. Second, the name is given according to the function. Third, the name is given according to the source. These provide us a good way to name enzyme, but there were some problems. What are these? What were these problems? The first one, here I have mentioned different enzyme had to go with the same name. The first problem is that we had, at that time, we had different enzymes, and all these enzymes have to go with the same name. For example, you have enzyme A, enzyme B, enzyme C. These are different enzymes, and by that time all of these different enzymes, they were given the same name X, let's suppose. That was a problem. This enzyme must be given a separate name, and that enzyme must be given another separate name, and this enzyme must be given another separate name. But because of this system, the different enzyme had to go with the same name. This was a problem. Yes, of course. That's the first problem. Second, the same enzyme had 2 or more names. Now this is another. Now you have one enzyme X, let's suppose. This is one enzyme X, any enzyme. Some people call it A, other people call it B, other people call it C. Because for one person this may be the function. For other person this may be the source. For other person this may be the ASE. So different people from the different school of thoughts, they would give what? They would give same name. They will give different names to the same enzyme. This is the same enzyme and different name was given. This is again a problem. We don't want this one and we don't want this one. We want that every enzyme should be given a proper name. So as a result of this, in order to avoid this issue, another system came. Now what is it? The need for a proper way of classification came into us. International. This is not international, this is international. International Union of Biochemistry, this is called it IUB. It's a kind of commission. Some scientists from the biochemistry, they came together, they sit together, and they thought that we should provide a proper system to name enzyme, because we don't want this sort of system, because with the passage of time a lot of more enzyme came to us. Many enzymes were discovered, and the enzyme reached to a very high number, and every enzyme must be given a proper name. If the enzymes were 5 to 10 or 20, it's okay, we would go with this system. But with the passage of time, the number of enzymes increased to a very high number, which is a very large number. So we must give a proper system. We must give proper name to each enzyme. Therefore, this committee, the committee of biochemists, the committee of scientists, they introduced a system called systemic 4-digit system. The 4-digit system is an advanced system to classify enzyme. The 4-digit system. In this 4-digit system, you will not face any problem, you will not face any confusion, you will not face these two problems. Different enzyme, same name. Same enzyme, different name. This problem, you will not see these problems. So what is this 4-digit system? In this 4-digit system, every enzyme is given 4-digit name. The name of every enzyme will be in this 4-digit, and what this 4-digit will show to us, I will explain it with just one example. Before I go ahead, before I proceed, there is an old video of mine that I have recorded on the classification of enzyme, that is ethyl system, oxidoreductase, transferases. These are 6 categories, 6 class, you can call 6 classes of enzyme. Throughout the world, you may have thousands of enzyme, different enzyme families. All of these enzyme, they must be from any of these 6 classes. So these 6 classes is called the ethyl system, because O is for oxidoreductases, oxidoreductases, Oxidoreductases. Oxidoreductases is not the name of single enzyme. It is a big family. It's a big family. Lot of enzymes will be there. Then transferases. It is not a single enzyme. It is the name of a very big family. Large number of enzymes are found here. Then hydrolysis. Then lyases. I have explained these names in that video. Please, if you want to go there, you can. But I will only explain today this transferases in today's lecture. Lyases, isomerases, and ligases. These are the 6 classes, the 6 families. Okay, now you just notice here, oxidoreductases is given the digit 1. Transferases is given the digit number 2. The hydrolysis digit number 3. Lyases digit 4, 5, and 6. Now, whenever you see 2, 3, 4, 5, 6, then this 3 means this hydrolysis. If you see this number 3 in the name of enzyme, I will explain. Okay, you just wait. There is one reaction where ATP and glucose, these 2 substrates are going to react with each other, and as a result of the reaction, ADP will be formed and a product glucose 6-phosphate will be formed. This is a chemical reaction. In this chemical reaction, the enzyme is also taking place. There is an enzyme which is catalyzing this reaction. If you notice here, what is happening in reality, from this ATP, 1-phosphate is transferring from the ATP to the glucose, is transferring from ATP to the glucose. And what is that group? This is the phosphate group, is going from ATP and towards the glucose. Now this is a very simple, you can see here the reaction. Now by the help of which enzyme this reaction is taking place? This is the enzyme. I will write here enzyme. E. This is an enzyme. The name of this enzyme is ATP glucose phosphortransferases. Glucose phosphortransferases, because this enzyme will transfer a phosphate group from ATP toward the glucose. This is just a simple name, but the 4-digit system for that enzyme is the 4-digit enzyme commission number. It is also called EC number, enzyme commission number 2711. Every enzyme name must be in 4-digit. I repeat, हर enzyme का नाम 4 digit में होगा. Every enzyme's name must be in 4 digit. This will be called the first digit. This is second digit, third and fourth digit. Every digit has some points to share with you. Every digit has some meaning, has very important potential meaning inside it. I will tell you, the first digit, whenever you see the first digit, this one, the first digit, whatsoever number it is, it may be 1, it may be 2 or 3 or 4 or 5 or 6, it may be any number. The first digit always show the class. You know, there are 6 classes. So the first digit always show what? The class name. So here is the class name. Here you can see it is 2. This is what? This is 2. So 2 means class name. Now this means that this enzyme is a transferase. This enzyme is what? Transferase. If there was 3, if there was 3, then that enzyme would be hydrolysis. If there was 4, then that enzyme would be lyases. If there was 5 number, then that would be isomerase. So this time that enzyme has given number 2. The first digit is 2. So it means this enzyme is from the transferase. Okay, fine. Then the second digit is 7. Now what does this digit show to us? The 7 shows the subclass. This shows the subclass. Means in the first digit we saw that the enzyme is transferring something, but from this digit we cannot deduct which group is transferring. Okay, from the first digit we just came to know, okay, it is transferring something, but we don't know what group, what thing, what is that atom or molecule that is going to transfer. We don't know, but the second digit will tell us that this group will be transferred, this group will be transferred. So the second group, the second digit is for subclass, class and subclass. So class name transferase 7. This 7 shows what? Phosphotransferase. Phosphotransferase means this enzyme is transferring phosphate group. If it is transferring phosphate group, it will be called phosphotransferase. And it may be if the enzyme transfers something else, like methyl, if it is going to transfer methyl, if it is going to transfer amino transferase. Okay, so if that group which is transferring was amino, then it will be amino transferase. But in this reaction, it is phosphotransferase. It is the phosphate group that is over, so we call it phosphotransferase. So for phosphotransferase, that is 7. This number may also vary. This number may get 6. This may be 5. This may be 4, 3, 2, 1. So this number will tell us what is the subclass. Means either it is phosphotransferase or amino transferase or something else. So the number will vary according to that enzyme. So this is the second digit. Second digit is for subclass, for class, subclass. And now the third digit tell us what? The third digit tell us, and this is the third digit, 1. Phosphotransferase with OH as acceptor. The third digit tell us that, okay, you have the enzyme. Okay, this enzyme is involving the transfer of phosphate group. Okay, this is okay, fine. Now they say that what is the group on that enzyme? What is that functional group on the enzyme with the help of which this enzyme transfer phosphate group? In order to transfer phosphate group from ATP to the glucose, the enzyme have what? The enzyme have OH group. This OH group belongs to the enzyme. This OH group belongs to the enzyme, and because of this OH group, that will accept what? That will accept the phosphate group from the ATP. So because of this OH group, we will give the name 1. Look, at the surface of enzyme, maybe SH group comes. So if the SH group comes, then the number will also change. This time that number, the third number, which is 1, indicates that there is OH. So phosphotransferase with OH means this is the enzyme. It contains OH group as acceptor. Means with the help of this OH group, it will accept the phosphate. Okay, fine. So now we got this. That this tell us the mechanism, how it happened, what group this enzyme have as acceptor. Then the last one, the last digit is again 1. The last digit tell us that, okay, you have the phosphate which is going to transfer. Now this phosphate, we know that there is phosphate and it is transferring, but we don't know it is transferring to what. The phosphate group can be transferred to glucose, the phosphate group can be transferred to fructose, the phosphate group can be transferred to any other molecule. So we must indicate it. So there is another number, right? There is another number that is usually called, we call it the substrate. Means this is our enzyme. It is going to act on the glucose. This glucose is a kind of substrate for it. In other words, the phosphate group is going towards glucose. So this number tell us what? This number tell us the glucose. This number may also vary. This number may be 2 or 3 or 4 or 5. If this is 1, this means it's glucose. If this number was something else, let's suppose that the phosphate group is transferring from ATP to glucose, but this time not to glucose, this time towards amino acid. If the phosphate group is transferring to an amino acid or some other compound, some other molecule, then the number would not be this one. The number would be something as some another number. So it may be 2 or 3 or 4. So the whole mechanism of the enzyme, this one enzyme, is in this 4-digit block. Now with the help of this 4-digit, we can tell that this enzyme belongs to the family transferases. Within this family, the phosphotransferases, and the 3rd digit tell us that this is the enzyme contain OH group, and the last digit tell us that that phosphate group is going to the glucose and not to other molecule. So this is a very special system. This is very unique special system to classify the enzymes. I hope that this lecture, you got this lecture very well. I can just tell something as is in a nutshell, that what we discussed today. We discussed today the classification or the naming of enzyme. For the classification or naming of enzyme, there are 2 ways. Number 1 is the improper way. You can call it the old ways. In the old way, enzymes were given names in 3 ways: by adding the suffix ASC to the substrate or the activity, by giving the name according to the broad function, and by giving the name according to the source. By these 3 ways we can give name to the enzyme. But there were some problems. Some problems were encountered. Some problems came to the ground by these type of naming system. In order to avoid the problems, there were some problems. In order to avoid these problems, we had to go with IUB. IUB is for International Union of Biochemistry. In this system, we introduced a 4-digit system in which the complete characterization of the enzyme is explained within this 4-digit system. In this lecture, I gave just one example of that enzyme, and you can also find the 4-digit number for other enzyme as well. You are an advanced level student, so I hope you got this lecture. Inshallah, see you in the next lecture from the same chapter. Take care and Allah Hafiz.